why and how this neurovirulent virulent simplex virus behaves so differently in foreign hosts versus natural hosts. Some of these studies result in the first diagnostic assay that enabled immunologic differentiation of B virus from HSV-1 and HSV-2, and subsequently the first complete sequence of the virus, of the B virus genome. And then she's also done a lot of additional things. All that being said, I'm sure that all of you who work with macaques in research can appreciate Dr. Hilliard's work with cercopithecine herpesvirus or B virus, and myself included at one time working with macaques. Knew her work very well, and I'm sure many of you know her work very well as well. We're very happy to have Dr. Hilliard here, and without further ado, I'll let you go ahead and give your lecture.
Thanks, Ken. Thank you. I can't thank you enough for this wonderful honor and invitation being here makes me feel. And what I'd like to do is tell you, let me back up a little bit. When Ken first called me, I had a talk that I was all prepared to give. And then I got to San Antonio yesterday. And I listened to Steve Bartol's talk. And I went, oh my God, they want more of a personal talk. And so a lot of this is revamped very quickly. And of course, I was here without too many pictures, but that brought to mind immediately, I had talked with Mike Hart, who is the chief veterinarian at Georgia State University, and who I'm sure all of you know. I asked him for some insight into what Russ Lindsey was like. And he said that when he first came to University of Alabama to do his fellowship, he arrived in Birmingham with his wife and newborn daughter, Lindsey. And Russ immediately said, oh, you have to stay with my wife, Ann, and I while you get settled. And I thought, well, that's kind of a nice story, you know. And his comment was, you know, with your daughter named Lindsey, you're going to have to stay with us. And so he commented on how hospitable and nice and wonderful Russ and Russ's family were, and just had a wonderful, wonderful time. And then I watched Steve, thinking about that, and I watched Steve's talk, and I thought, oh, I get it. Russ, Lindsey, yeah, they want to know something, you know. So I'll tell you the next connection. The next connection was Mike said, when he finished the fellowship and he got his first job at Auburn, Russ came to visit with him. And he happened to be out of his office that day. And Russ came to his office, and it turns out that Mike was off coaching his son's Little League team. And Russ left him a note, and in my field, that note would say, where the hell are you? Why aren't you in the lab? But Russ's comment was, I'm so glad you put family first, and that's the way it should always be. So then it really made sense that Russ was the kind of guy that would really want to know each of you personally, and what you do and what you're like. And so giving this as more of my personal journey made much more sense. So it was like the final pressure cooker of knowing what content I was going to share with you today emerged into, I think, what was important to Russ, which is us as people, and a compassionate look, as Sally said, really prevailed in his mind. He was certainly a giant, and he was a man with few pictures, as you can see. You search, you search the internet, and what you will see is there are no pictures of Russ. So I think he was probably a very self-effacing man, too. I served on several committees with him at NIH and on site visit teams, and he was just the most elegant and debonair of gentlemen, and certainly with great Southern hospitality that Mike attested to with his family, and a man that is certainly, in my community, a silverback. So I'm very honored to speak at his memorial lecture. So with that said about Russ, I think one of the next things we remember about Russ is that his contributions to understanding mycoplasma are just giant. And that's just one of few of his contributions. But again, in the research laboratory, mycoplasma is a big thing for us as well as you guys in the lab animal facilities, because mycoplasma can really destroy, much the same way gut microbiome, and Craig's wonderful talk described, gut microbiome can certainly impact your science. Mycoplasma is horrific when it gets into your cell cultures. And I can't even imagine a 3D or a 2D cell culture model that's quietly infected with mycoplasma, and the nightmare that that can cause with the data that we generate. So moving on toward that personal look, my students in my graduate seminar course always want the speakers to start off telling the students a little bit about themselves. And the range of responses from the speakers when I tell them this, they go, "Oh, my, no." And to, "Oh, like what?" And I explain to the students, scientists generally are pretty introverted and insecure and beaten down. And so they don't really want to talk about themselves. And that's the way I felt when I was preparing this. And listening to Steve's talk, one of the first things he said to you all was, "Wow, this is really hard talking about yourself. This is," and it is very, very hard. So I hope you will forgive me as I fumble through. But I did, in preparing the inserted slides, I did learn a lot about myself. One, that, wow, I'm really old. That wasn't really flattering. And two, I shouldn't be up on this stage with the company that has preceded me for this lecture, because they've had very gentrified educations, and you'll see I wound my way through life. So I'll start with when I was in high school, because I think that's, and I'll give you a look a little further back in a minute, but in high school, when I hit high school, it was time for science fairs, and my first science fair I did the solar system, which was a big flop, as you might imagine. The next year, though, I decided to go and discover antibiotics in the dirt in the backyard. And that proved to be a big winner and got me to be the state of Louisiana delegate to the International Science Fair. But I have to tell you a funny story given Craig's talk. My animal model was a hamster. More than one, but not a handful. And the only place a high school student could get in 1965 hamsters would be the dime store, and we call them 5 and 10s. I don't know, some of you will remember these. So I went to the dime store, and 5 and 10, and all they had were these 2 male hamsters that were fighting. And I said, "Well, okay, I'll take those." And I brought them home. This was before IACUC. And so I do have an appreciation for the benefits of IACUC now. So I put these guys in a box with a light bulb, so they were probably sweating like all heck, but to keep them at a warm temperature. Yeah, well, it was the South. So toward the end of the experiments of curing them of these infections that I gave them, one got loose and went into the framework structure of my parents' home after he bit me. And the bad news is I was young enough and stupid enough to wonder if I was going to die of rabies in 2 weeks first. But the second one was, he's going to stink up the walls of my parents' house, and then they're going to know that I wasn't going to tell them for sure. I didn't plan to tell them I was going to die from rabies in 2 weeks. And I didn't plan to tell them about a hamster being trapped in their house. So that was my first big foray into scientific investigation. And Sally's 3 R's reminded me that at the very last minute, my father said, you know, for the title, you should put the SP 3 R's. I don't remember what the SP stood for, but now I have an idea what the R's may have stood for, so thanks to Sally. Must have been something, you know, that was pithy, and I'm sure I remembered at the time, but it was the first opportunity to fly on a commercial airline, to go to St. Louis, to go to the International Science Fair, and when I got there and I saw people's projects, I just about freaked out. Mine looked so podunk. But it was fun and it was a great experience, and I followed that project for the next 3 years. What were the benefits of that? Well, I found out at the science fair that everybody had a job at their local universities, and that their universities probably had done their projects because they were really sophisticated projects. But I got a job as a lab dishwasher at LSU in the department, no less, of bacteriology. Oh, and it was in the basement of an old building. And I thought that that was just the cat's pajamas. I just thought that was the coolest thing that could have ever happened. When it came time to start at the university, I was only 15 at the time that this was all happening. And when I started at the university, I thought, now, what do I major in? I guess I could major in psychology or something. I had no idea that you could major in something that would let you work in the lab all the time. So I wasn't the brightest of kids, nationally speaking. So the guys in the lab said, why would you major in psychology? Why don't you major in microbiology? And I said, you can do that? Wow, that is awesome. And so I've pretty much never left the lab, but as a sophomore, the chair of the department, Marian Sokolovsky, called me in and he said, Julia, we're out of money. And actually he said, Kathy, we're out of money, because his daughter's name was Kathy, and he just decided that since my middle name was Kathleen, I would be Kathy. So he said, Kathy, we're out of money. We can't continue your job. I mean, 50 cents an hour, really. And I said, okay, you can't continue, but I got to work in a lab. And he said, no, you don't understand. You can't. And I said, no, but I have to work in a lab. And I said this about 3 or 4 times, and he looked at me like, are you deaf? And so he picked up the phone and he called Dr. Virginia Williams, who was working on metabolic pathways in bacteria using bioreactors in the chemistry department. And I got a job in her lab. And by the time I was ready to graduate, she was insisting that I needed to go to the new UC School of Medicine and continue. And then she unfortunately passed away. So I lost the value of her direction and went off in another course. But she was an awesome mentor, and I really learned a lot about research technique. Now, the next picture I'm going to show you is the earlier picture I referred to. I'd like to get it on the screen and off as quickly as possible, but I wanted to emphasize to you all that I have had an ever-abiding love in science and medicine, and you can see from the earliest days that I was well-fed, and my career choices were lab animal medicine. You can see the rabbit there. I did use a lot of rabbits in my research eventually. Antibody production. Would I be an experimentalist? An animal models expert. That rabbit really says a lot. Or comedy. So I guess you see that, the first part of my life is comedy, and the second part, I finally found a home. This is my cat's reaction when he saw that picture. I was working at the kitchen table making this presentation, and the kitten said, "Oh, what? Did you just see that?" So I grew up, and this was me at Baylor College of Medicine. I'm the middle person. In 1973, I noticed Steve showed pictures, so these are the only pictures I have. But at 19, I went to Berlin, West Berlin, because the wall was still up, and I worked in a typhus lab, and they said, again, this has got to be comedy. They put me on the stool samples. And they said, if when you pop the cork, you get something on you, stand up and say, "Stuhle, Stuhle." It's like, that's very embarrassing for a 19-year-old, And fortunately, it only happened once, and I don't think anybody else in the lab said that. I think it was just a joke on me. But I was definitely a confirmed laboratorian by this point. However, in my senior year, I took immunology from Dr. Seifert, and who locked the door at 8 a.m. and nobody else could come in, so you weren't late. But I was really bitten by immunology, and I graduated a little early, or was scheduled to have an empty semester, so I did an internship at Argonne National Lab, where I accidentally had my advisor's lab completely shut down due to radioactive contamination. So he came in, he was a very meek guy, and he came in the next morning, he goes, "Julia, what happened last night?" So I know I had my arms scrubbed, and they went and checked my clothes in my dorm room, and it was a big deal. But I learned how to work with P32. So then I went to an internship at Baylor College of Medicine, and I was really going to go off to University of Illinois to study with Sheldon Dray in my dream field, immunology, and my advisor said, every scientist really needs a strong biochemistry background. And I said, yeah, but this is a pharmacology department. He goes, no, this is, we do biochemistry here. I said, okay, well, yeah, I'm kind of tired of moving around by this time. So I stayed there, and I absolutely hated the project, but I did become a Ph.D. student. And I'm going to show you that this became my home for the next 7 years, both as a graduate student and as a postdoc. And my first, as a postdoc, I moved over to MD Anderson, and I worked with Dr. Ellen Ritchie, who taught me so much about B cells and T cells, and I'm forever in her debt, and I think she's still there with her lab. But I wanted to show you, these were my PhD studies, and back in the turn of the century, you had to type on a typewriter, and you had to use, no page could have any typos. And so one typo, and you had to start that page all over again, and you can see how freaky these look. It's just, I was studying repair methylation in Novikov hepatoma cells, and I try to jazz it up now and say, I was studying cancer and DNA methylation in specific cancers using, these mouse cells. I could, maybe convince my parents of something like that, but repair methylation of parental DNA and synchronized cultures of Novikov hepatoma cells was not what was thrilling me. But I have to say, I learned how to do these density gradients really, really well. And then we discovered, after we published all this data, that the whole thing was fundamentally an artifact of the cell synchronization methodology I was using. So they still gave me my Ph.D., so that was great. The chair of the department said, this is really not a pharmacology experiment. I think you need to go back and do at least one drug study. So I put caffeine in the system. I've had a long love of caffeine, having started drinking it at 4. Truly, that's what they do in Louisiana. It makes us look a little sharper. Sorry, I'm from Louisiana, okay? So if anyone's from Louisiana, don't take this wrong. So I went back to my first love, which was immunology, and up 6 floors was one of the most famous virologists at the time, Joe Melnick. And Joe said, I have a perfect lab for you. You're going to go in with Florence Farber and you're going to study DNA remodeling. And I said, no, I don't want to do that. I really came up here to study immunology and virology. No, it's a good fit based on your DNA methylation studies. No, I don't want to do that. So he finally put me with Gordon Driesman, who was working on hepatitis. And he didn't think that I should join the hepatitis group, so he let me do my own version of research with HSV-2 and looking at AG4 as a novel viral antigen. And so that began my foray into sexually transmitted diseases, which of course B virus is one for monkeys. But I knew a lot about the family of viruses, so that was cool. And then I went on, as I said, to Ellen's lab, and I had gotten really interested in cell surface antigens, first with HSV-2 and looking at what the virus puts on the surface of a cell and how that affects the host immune system. Some really cool stuff was happening. And then I started looking at PBMCs and purifying T cells, and at the time, we had no fax machines, so you used sheep red blood cells to rosette out the T cells. I don't know if anybody remembers those techniques from those days, but I started looking at, I was an expert at radio labeling again, I-125, all the proteins on the surface of the cells, and right across the hall was a young investigator who had just come, and he talked to Ellen, and they got to talking, and he started working on the surface of cells too. And his name, by the way, is Jim Allison, and he won the Nobel Prize a couple years ago. So I can at least say I was close to the lab where Jim started. But the benefits were I really became good at purifying proteins and studying immune responses. And what I didn't realize is that at Baylor, when you work with viruses, you could just go up to this guy named Eddie Seidel and say, Eddie, I need some HSV, and he'd hand you a vial of HSV. And we didn't wear gloves. We didn't even have biosafety containment cabinets. We had static boxes. And so that was a real transition from working with mouse cells in culture. But when I took my first position, I soon discovered I knew nothing about growing viruses because Eddie had given me all the viruses. So I was calling him frequently when I moved to Southwest Foundation for Biomedical Research, which now is Texas Biomedical. And they said, well, we have this problem with B virus. This was 1980, so wasn't much of a problem. Now, cases hadn't really happened since 75 at UC, University of California, Davis, where I think it was Davis, one infection had occurred, and that was studied pretty intensely. But it was interesting because the chair of my department at Baylor called and he said, I used to work on B virus. Well, I think it's the first time he had ever talked to me. I went through a whole fellowship there and he didn't say too much. other than offer me a position, which I naively said, no, I need to go learn more immunology. So I turned down that position and ended up surreptitiously at the Southwest Foundation, where I was introduced to all kinds of new things. And what I first realized is that the literature was sparse, but very, very informative on B virus. And I knew B virus was very much like HSV-1 and HSV-2, but I didn't know too much more than that. And I thought I would focus on learning more about the virus. So the virus had been discovered in 1929 when Bill Brebner got infected. He was a young MD doing a necropsy on an experimental macaque. And his co-workers, Gay and Holden, isolated the virus, but didn't have the virology background to really study it. So just like today, when you have a young investigator studying something that looks really cool, you always have a more mature investigator that goes, oh, let me help you. So I don't think Gay and Holden's name was mentioned in the discovery of the virus, but Albert Sabin published the first paper and called it B virus after Dr. Brebner. Really bad nomenclature, but it stuck. And so now most people think outside this room and in the community at large that I work on hepatitis B, and they can't understand. I get a lot of calls from emergency departments at hospitals that they have a person who's been bitten by a macaque that may now have hepatitis B. So I calmly explain. But going through the literature, there were about 18 cases that were described. And the trend seemed to be that when we as a community, as a biomedical community, import macaques, that's when we start getting high population, high census numbers in our institutions, that's when we start to see B virus cases. And so this was the this was the middle 1980s. And it was very clear that with the importation of macaques under really terrible conditions, much like my hamster experiments were under terrible conditions, these were worse. The importation of macaques, they were crammed into boats, cargo ships, and by the time they got to the U.S., half of them were dead. And these were the animals that we made kidneys from to grow poliovirus in culture. And so I don't know if you're aware, but we check a lot of kidney cells from macaques because frequently after 2 or 3 passages in the 80s, the culture would break out with B virus. So we started checking, and about this time with the deaths that were happening, things changed with respect to the way we thought of macaque monkeys and the way NIH approached them as the invaluable animal model that they were. So back then, the primate centers were not national treasures, they were regional treasures. So it was the California Regional Primate Research Center that had a case that was not documented widely. And that was an interesting case because it was the most recent serum samples that we could get a handle on. And then there was a Vietnam vet that was bitten while he was in a bunker in Vietnam, and he was diagnosed by the Army to have varicella zoster. So we discovered him, and that will emerge in a moment, in a little bit. But basically, from California Primate Center, a bunch of bonnet macaques got sick, and Rubner and his colleagues isolated the virus and grew it in culture, and these were some of the first pictures that we saw from these animals because microscopy and photography had merged to a point where they were a lot sharper. not so sharp appearing here, but that was really sharp for the day. But you can see that there's a mass of virions that grow and assemble, replicate and assemble in the nucleus of a cell. And one cell can carry anywhere from 1,000 to 10,000 particles. And moreover, if you look at the cells at a lower magnification, you'll see that B virus has this wonderful ability to fuse cells that it's going to infect. And so it's a lot like respiratory syncytial virus, and you get these big, ugly, I think it kind of looks like science fiction there, you know, it's just these big, ugly, polynucleated cells that are all fused together. And I tell you, the first time you see this picture in your lab, or the first time I saw it in my lab, it was like, oh, that's really a nightmare CPE, or cytopathic effect. And about the same time, we had worked out that it replicates really fast. Now, the replication curves on the right demonstrated that it had about an 18-hour life replication doubling time, and that almost a log less virus was found in the supernatant fluids. So most of the virus was held in these cells that had fused together. And that didn't make sense till much later, and I'll share the reasons for that in a moment. But for those of you that haven't heard too much about B virus is the herpes simplex of macaques. All of us, all species have their own simplex viruses. And herpes viruses have what we call alpha, beta, and gamma viruses, as you all know. And these are alpha herpes viruses. And in alpha herpes viruses, there's two types of herpes viruses. There's the varicella-viruses and the simplex viruses. And so my focus became on the simplex virus family, subfamily. And so B virus in macaques, no big deal. It's like HSV in humans. Little like HSV-2 as well as 1 combined together. And HSV-1 and R2 don't cause much damage in people, except maybe a little social stigma if you have HSV-2. And in fact, what I've learned over the years is people would rather have B virus than HSV-2. So it's still apparently a big social stigma. So, but I'm not sure why, and I'll tell you that in a minute too. So, but when a person contracts B virus, the results are deadly. And in putting this talk together, I had this great idea, and hopefully I'll tell you about it in a couple years. I think I know now why, but I have some experiments to do. But at any rate, in people, this is about an 80% mortality. And when I joined the lab at Southwest Foundation, there was really nothing that could be done. Harold Zwartow at Porton Down had discovered that acyclovir might be of some benefit in rabbits, but if you took them off of acyclovir, they died in 2 weeks. So it looked like if we saw another patient, we were going to need to treat lifelong. But the interesting thing was, and for those of you that might run HSV-2 vaccine trials, is they're all done in macaques. And we've known for a while, but that may not be the best animal model. So looking at the 3 Rs with data that I think we'll soon publish might suggest that they're not the very best animal model. The Southwest Foundation had a BSL-4, and it looked like this. This was all submarine equipment that had been assembled to make these glove boxes. And the only other labs really that had these were in Novosibirsk, and I wasn't going to go there, and CDC. And I chose the Southwest Foundation and spent the next 18 years working in this box. Now after I left, they decided to make it really sophisticated, and it's a suit lab, and it's great, but I unfortunately left a bit soon. So my first project in foray was to really get in and purify the polypeptides and glycoproteins of the virus, and to see what its components were like, because that's what I had learned to do with HSV-1 and HSV-2, particularly HSV-2. And I learned, too, that the prominent means of getting infected if you're a macaque was when you reach sexual maturity. We studied how this virus looked at different times, post-replication or post-infection, and carried out these long experiments where I would frequently spend the night in the lab and learn about the polypeptides and when they appeared during infection. And so next we looked at how to differentiate the virus with a friend of mine who had postdoced also at Baylor. And he created ELISA assays that if we just use the right infected cell lysate with all the properties that I showed you in the previous slide, we could effectively differentiate B virus antibodies from HSV-1 and HSV-2 antibodies. Now, this is what plagued Albert Sabin. And Albert discovered that if you took serum from a person with HSV-1, it didn't do diddly to B virus. But if you took serum from a B virus infected person, it neutralized HSV-1 and HSV-2. And at that point in 1985, I thought, we do have a vaccine against HSV-2. Just get B virus because the antibodies will protect you. And that still holds true today. But I haven't found any cohorts to study this. So the next thing that happened, I don't know what I was doing. I was having fun in the lab again, and I was discovering all about this virus that we didn't know much about. It was a really great privilege. But then storm clouds mounted, and I got a call right after we published this paper from the Naval Aerospace Medical Research Labs in Pensacola. And they had two gentlemen that each had, three gentlemen actually, that had been bitten or scratched by a juvenile macaque. It was really an immature macaque. And both were now in the hospital, and the third one was not feeling altogether too well from little tiny things like this, just little healed up bites. That ring finger bite, scratch, from a cage scratch, from a jerry-rigged cage fastener, transmitted B virus to this gentleman. And a little tiny bite here on Mr. Steve Woodson's arm and on Larry Smith's finger, and this is what you end up with. So you end up with an amazing catastrophe of ascending paralysis, and first bladder paralysis, then diaphragmatic paralysis, swallowing goes, and even your ability to blink your eyes. And we know this because one of the most famous virologists, Carl Habel, who wrote Fundamentals of Virology, actually had not worked with macaques for 20 years, and in the late 80s got sick and lived as a total quadriplegic, only able to blink his eyes for the next 10 years in an iron lung. And so we learned a lot from these cases, and it was just amazing. And I have one more picture of myself. This was us dressing for the first fatality because we were able to get autopsy specimens in collaboration with the pathologists at Baptist Hospital in Pensacola. And that started off a completely different facet of my life. And so having undergone a smorgasbord of laboratory experiences and knowing I wanted to be in the lab, it's a good thing I knew I wanted to be in the lab because we've been in the lab ever since, usually at least 6 days a week, trying to figure this virus out. So what we learned from the next bam, bam, bam cases were that every patient demonstrated pretty much the same thing. And what we were amazed to find out is this virus invades the brain completely differently than HSV-1 or HSV-2. It just centers in the brain stem, and it results in the herniation of the brain stem and the loss of all autonomic functions. And most deaths, of course, are due to respiratory paralysis. And so it's really interesting. You can, now we've discovered, intervene successfully, but we've then discovered that in those people that we can intervene in, they can reactivate virus. And so they live with that consequence and have to stay on acyclovir. This was a retinopathy that became a bilateral retinopathy due to B virus. And this is a B virus infection that occurred in 1980. So he reactivated in 2004. So in the laboratory, what we were learning is the difference between this virus and HSV-1 and HSV-2, it didn't use the same receptors, which was amazing. But then as I started teaching immunology, I learned that that really is going to mess up the immune response, because normally in a herpesvirus infection, TNF alpha is upregulated, not necessary with B virus. So it's almost like it's a stealth virus. And so that makes for a really different set of symptoms that will happen. The virus gets in, it's, again, stealth invader. It suppresses all the pro-inflammatory defenses, and it doesn't have the neurovirulence gene that everybody claimed was the neurovirulence gene in HSV-1 and HSV-2. It completely lacks it. So HSV-1 and HSV-2 did not emerge from B virus. And it has another protein that is also shared with HSV-1, HSV-2, US11, and US11 actually is amazing because it's upregulated early in B virus infections, not late like it is in HSV-1. And I think that is a very important clue why this virus is so radical in humans. And again, look out for papers in the near future. So it uses stress pathways completely different. The cell's like not even stressed when it's infected. It's like nothing's happening. And it also goes ahead and it expresses antigens on the surface of the cell. HSV-1 and HSV-2 are at least clever enough to suppress this. But B virus got a different road. So we had all of these findings. We knew the polypeptides. We had made ELISAs. We sequenced the genome. We knew the relationship between all of the non-human primate viruses, and that if you don't have a simplex virus in a host, it's because you haven't found it yet. And we had done many experiments with different antivirals and found a secret weapon that nobody wants to use because they feel like it's too toxic of a drug. So my pharmacology training came back, and we could use this toxic drug at thousandths of a dose that it was toxic at, and it completely just completely inhibited B virus replication. So hopefully someday we'll be able to try that if we have an unfortunate zoonotic infection again. And now we've discovered and identified epitopes that can be used, so I hope in a few years you can do your own assays in your own labs using these epitopes and won't have any need for growing the virus. And along the way, we had some fun in the lab. We discovered a silverleaf Langer virus, a Pseudomongoby virus, the baboon simplex virus, which we all thought back then was SA8, turned out to be a virus of its own, and a reovirus that was a combination, a recombinant virus between rattlesnake and birds. And that was fun. And co-discovered the chimpanzee virus. all of these simplex viruses. I couldn't have done this without a lot of help along the way. I've had an adventure, it seems trite, adventure of a lifetime, but man, it's been an adventure from needing to be in the lab to learning all of these things. And I think it's just been amazing to teach students about this virus and about the immunology of the host system. And I couldn't have ever gotten started without Leah Whitehair telling me how to write a grant. So it's been amazing, and mostly this community has just been beyond awesome to work with. I've learned so much from you guys, and it's been a privilege to work with all of you. And I hope this little odyssey does honor to Russ. That's kind of maybe what he wanted, but it was an adventure preparing it too. And so I'd like to thank all of you, and let's give a round of applause to Russ. If anybody finds a good picture of him, I wish you would send it to me. That would be great. Thank you.